Good afternoon, everybody. My name is DEON De Jager, and I'm from the University of Pretoria Molecular Ecology and Evolution program in the Department of Biochemistry, Genetics and Microbiology. Here's my face. For those of you who didn't manage to see it earlier, you get another opportunity here. And this project is a collaboration between us and our colleagues at the Center of Excellence for Biomedical Tuberculosis Research at the University of Stellenbosch. So this is the first population genomic study of African buffalo, and we sequenced 40 genomes at low coverage, approximately 7 times, and these 40 genomes are from 4 protected areas in South Africa. So we sequenced 15 from Kruger National Park, 15 from Khloe and Mfolozi Park, 5 from Addo Elephant National Park, and 5 from Mokala National Park. Okay, so just a bit of history on the South African populations that we sampled. So in the 1890s and early 1900s, 95% of the buffalo population in South Africa and much of Southern Africa died due to disease epidemics, predominantly rinderpest, as well as overhunting. And if you want to find out more about this rinderpest outbreak, give a listen to This Podcast Will Kill You. It has an episode on rinderpest and what devastating effects it has, a bit of its history and so forth. So really interesting. Anyway, after the rinderpest outbreak, hunting of buffalo and other ungulates still continued in order to eliminate these animals as hosts of the tsetse fly, which is a vector of nagana in cattle and sleeping sickness in humans. And so to protect themselves and the livestock, the wildlife was hunted. That was until about the time when these areas were declared as protected areas, at which point we assume that hunting ended or was at least drastically reduced. And then since being established as protected areas, neither Khloe nor Addo has had any introduction of buffalo from other populations or any recorded introductions. The Mokala National Park population, on the other hand, was established through a disease-free breeding program by San Parks to establish a disease-free population of Kruger buffalo with high diversity. So getting to the questions and methods. So we wanted to know then in this study, have the recent population bottlenecks resulted in low genome-wide diversity or high inbreeding coefficients? And did the disease-free breeding program create a founder effect in Mokala National Park? So a founder effect is when a population is founded by just a few individuals and thus grows from there and has low diversity because of only the diversity that existed within those few individuals. So to do this, we calculated individual heterozygosity and inbreeding coefficients, as well as estimated inbreeding tracts across the genomes. And these are measures of genetic diversity that can be much more accurately estimated with whole genome data as opposed to microsatellites. Okay, so getting to the results. Here we've got genome-wide heterozygosity on the x-axis and the samples on the y-axis. You don't need to see their names, just which population they come from. So we found genome-wide diversity was significantly lower in Khloe compared to Kruger, which we used as a benchmark in the study, and also significantly lower in Addo as compared to Kruger. Genome-wide heterozygosity was statistically equivalent in Mokala National Park compared to Kruger, thus indicating that the high genetic diversity in Kruger National Park was actually maintained in the disease-free population in Mokala, which is a really positive result for the disease-free breeding program. But how does this genome-wide diversity compare to other species, other mammals for which this analysis has been done? Well, in this plot here, we can see that Cape buffalo has the highest genome-wide heterozygosity of any mammal for which this type of analysis has been done. If we then look at the inbreeding values, we see that Addo jumps out as having really high inbreeding values, or higher than the rest. So inbreeding values are between 0.2 and 0.25, whereas Kruger, Khloe, and Mokala have really low inbreeding values, close to 0. So what does this mean? So inbreeding, so the offspring from the mating of first-order relatives, so for example, full siblings, brother and sister, the offspring of that mating would have an expected individual inbreeding coefficient of 0.25. while the offspring of second order relatives, so half siblings, would have an expected inbreeding value of 0.125, although in reality there is some variance around these values, so it can be slightly higher, slightly lower. But what this means then is the inbreeding coefficients estimated here for Addo would suggest that the parents of these individuals, and we can extrapolate to the population, were effectively relatives at the half or full sibling level. So that's really worrying for Addo. We've got one individual here from Kruger with a relatively high inbreeding coefficient and also lower heterozygosity than the other Kruger samples. And this individual was most likely a mating of closely related individuals, offspring of a mating between closely related individuals within a herd of buffalo. So that's expected to happen from time to time, given the mating structure and hierarchy and the herd life of buffalo. If we then look at the inbreeding tracts, we find that Addo buffalo has a lot of inbreeding tracts across their genomes. So the vertical blue lines here indicate an inbreeding tract, and the blocks here, so from left to right, is the beginning and the end of a chromosome. So this is all the positions along a chromosome, and then these are different individuals. Okay, and then this is just one chromosome, so there's 52 more. And yeah, so what we can see is the vertical blue lines show yes, there's inbreeding, and the white spaces show no inbreeding. So a lot of inbreeding tracts along the Addo buffalo genomes, and the Kruger sample here is just an empty plot, so no inbreeding coefficient. No inbreeding tracts. If we then look at a zoomed out view, so each sort of column here is one chromosome, so these are 4 chromosomes here, and we can see the Addo buffalo with all their blue vertical lines here showing inbreeding tracts. Whereas if we look at the Khlukhluwe samples, for example, shaded in grey here, we see that they don't have any inbreeding tracts and no blue lines there. And this individual here has some blue lines on this chromosome, but not on the others. And this is the sample from Kruger National Park with that relatively high inbreeding coefficient. So how do we then reconcile these results with the population history of each of these populations and the differences between them? So it can essentially be explained by the severity of the initial bottleneck, so the absolute number of buffalo that survived, and how long the bottleneck lasted, and also the subsequent recovery. So an important concept to understand here is that small population sizes, if you have a small population size for long periods, so multiple generations, this leads to a loss of genetic diversity through genetic drift and eventually leads to inbreeding. And this can be countered through natural migration between populations or translocations to bring in new individuals. So the initial population sizes, so the severity of the bottlenecks wasn't known as soon as the areas were declared as protected areas, but we do have estimates of the lowest population size in each population at some point back in time. So the rinderpest outbreak and hunting seems to have had a limited effect on the genetic diversity in Kruger buffalo, if at all, because of the sheer number of buffalo that survived, so more than 1,600, and all these buffalo then harbored most of the genetic variation that was present in the population before the die-off. So then also hunting pressure was likely less severe due to the presence of malaria in the Kruger National Park, so reduced human activity. And Kruger National Park is also connected to large neighboring natural areas in Zimbabwe and Mozambique, and thus natural migration between populations between these areas also would have increased genetic diversity after the disease outbreak. But given the similar lowest population size of Khlukhluwe and Addo, how can we explain the differences in genome-wide diversity and inbreeding between these populations? Well, Addo was declared as a protected area a full 35 years, so that's 5 to 7 buffalo generations after Khlukhluwe. So hunting of buffalo in Addo continued for much longer than it did in Khlukhluwe, and it was also most likely more intense in Addo compared to Khlukhluwe, because in Khlukhluwe you had the presence of the tsetse fly, which also meant less human activity and cattle husbandry. So not only did hunting continue longer in Addo after the bottleneck, but it likely started even earlier as well, with colonialists who did a lot of the hunting, settling in the Cape first before moving to other parts of the country. Additionally, Hluhluwe seems to have had a higher population growth rate after the initial bottleneck, as its population increased, is currently at around 4,500, but has gone up to 8,000 in the past. And a fast population growth rate means more mutations per generation, more new mutations are created, and thus higher genetic diversity results. And the same can be said for Kruger, which also probably had a really high population growth rate after the bottleneck and has a really large population now. Whereas Addo population has never grown to more than about 1,000 individuals, which may be due to regular offtake of buffalo in Addo or a lower carrying capacity in the Cape Floristic region with nutrient-poor soil and unpalatable plants in that region. Okay, so then what results from all this work? Well, first of all, we would suggest genetic supplementation of the Addo population to reduce the inbreeding as a matter of urgency. And the source for this supplementation should be Mokala National Park, as it has disease-free buffalo and high diversity. So the suggestion is about one breeding bull every generation, so as not to swamp the genetic identity and then the unique variation of the Addo buffalo with Kruger genetics. In Mokala National Park, we didn't detect a founder effect, but we suggest that genetic monitoring should be implemented, and that says maybe once every 10 years, evaluate the genetic diversity to determine whether there's been any loss, and then SANParks should be willing to supplement with more Kruger buffalo, disease-free of course, if required. A similar result or conclusion for Hluhluwe. We didn't find any inbreeding, but there was slightly lower genetic diversity than in Kruger. And thus we also suggest genetic monitoring of this population and supplementation if genetic diversity is lost or inbreeding occurs. For Kruger, there's not much to do as the large population more or less takes care of itself. With that, I'd like to thank my collaborators and funding bodies, and thank you for your attention.